Welcome to this video where we will discuss one of the most important biological processes that occur within cells: the replication, transcription, and translation of DNA. These processes are fundamental to understanding how genetic information is transferred in living organisms and how proteins, which represent life itself, are built. Part 1. DNA replication. The first process we will talk about is replication. This process is the way in which DNA is copied before cell division, ensuring that each new cell contains the same genetic material. 1. Initiation. The replication process begins when the two strands of double-stranded DNA are separated by a special enzyme called helicase. This enzyme unwinds the DNA to create an opening known as the replication fork. 2. Elongation. Once the double strand is opened, other enzymes such as DNA polymerase begin to add the appropriate nitrogenous bases, such as adenine with thymine, and cytosine with guanine. 3. Result. Eventually, two identical strands of DNA are created, and this occurs accurately so that each new cell can obtain a copy of the original genes. The replication process ensures that genetic information is accurately passed from generation to generation. Part 2. Transcription. After the DNA is copied in the replication process, the transcription process follows. At this stage, the information from DNA is converted into RNA, and this process occurs in the nucleus. 1. What happens during transcription? During transcription, a portion of the DNA is opened by RNA polymerase, the enzyme responsible for copying the information. 2. Formation of mRNA. After opening the DNA, RNA polymerase adds RNA bases, such as adenine with uracil instead of thymine, and cytosine with guanine. mRNA, messenger RNA, is created, carrying the genetic message from DNA to the ribosomes. 3. The result. After the transcription process is complete, mRNA is formed, containing a copy of the gene that needs to be translated into protein. Once mRNA is copied, it moves from the nucleus to the cytoplasm to be translated. Part 3. Translation. After mRNA is created, we move on to the translation process, which is the process through which the genetic message is converted into proteins. Proteins are responsible for all vital activities in cells. 1. How translation occurs. mRNA moves to the ribosome in the cytoplasm. The ribosome is the place where the protein building process takes place. 2. Reading the information. Inside the ribosome, the codons present on the mRNA are read. A codon is a sequence of 3 nitrogenous bases that represents a specific amino acid. 3. Bringing in amino acids. tRNA molecules, which are small molecules responsible for bringing specific amino acids based on the codons in the mRNA. 4. Arranging amino acids. Amino acids are linked in a long chain to form proteins. Proteins are essential molecules that perform all functions in living cells. This process occurs quickly and accurately and is considered the foundation for all vital processes in living organisms. Part 4. Conclusion. So, to summarize everything we talked about: replication, copying DNA to ensure that each new cell receives the same genetic information. Transcription, converting information from DNA to mRNA. Translation, converting mRNA into proteins that form the cells and organs that make living organisms function. Each of these processes together forms the foundations of life and helps in transferring genetic information and building the proteins necessary for body functions. By understanding these processes, we can understand how changes in DNA affect the health of living organisms. At the end of the video, don't forget to subscribe to the channel and activate the notification button so we can explore the science of biology together. Thank you.